Good afternoon, everyone. Welcome to the 2 o'clock briefing, using mass spectrometry to unmask clues from the past. We have 3 speakers on our briefing today. They are Ruth Ann Armitage, Professor of Chemistry at Eastern Michigan University, Christina Warner, Professor of Microbiome Sciences at the Max Planck Institute for the Science of Human History, and Elena Schrader, the Arnold O. Beckman Postdoctoral Fellow at North Carolina State University. So we'll start our briefing now, and we'll have a hard stop at 2:45. Thank you so much.
Thanks so much. So my research group uses ambient ionization, specifically direct analysis in real time, or DART, mass spectrometry, as a fast way to identify small molecules without having to do any sample preparation. So we specifically apply these methods to characterize art and archaeological materials that, because these are rare and precious artifacts, we want to preserve them, and so we really can only use very tiny samples if we take samples at all, and this is all we really need to do DART. So by studying ancient materials, which you'll hear over and over again from all of us, I'm sure, helps us understand something about people in the past, their technology, their trade, things about what they ate and drank. So this is some of my students doing some DART in the laboratory. And much of what I'm going to talk about tomorrow is our work on identifying dyes in textiles. So we use a tiny, tiny clump of fibers from just a yarn, and I had a little demo that I forgot to pull out, but it's just a tiny fleck of material that we can hold into the gap that you see here between the blue ion source and the rest of the instrument, and we get signal instantaneously. So this is really a different way of doing this. So this is the kind of sample that we get to use. So we've been studying South American textiles quite a bit. And so you can see that from the sample that we get, we have a very small little area that we take a sample of, hold it into the mass spectrometer, and we get signal for, in positive ion mode, we get indigo, and in negative ion mode, we can see better the red colorants that are present. Now, the other story that seems to be getting a little bit more attention is our work where we were asked by ceramic specialists at Ferry Farm, which was George Washington's boyhood home, to look at the composition of glue residues that were found on these 18th century ceramics. And these would have belonged to the Washington family. So there are so many different 18th century recipes for homemade glues, but I'm really only going to talk about 2. And one that was considered to be really great for repairing broken ceramics was actually made of grated cheese, milk, and the kind of lime that you would use to make mortar. And this made sort of a cheese mortar-like material. So we did some DART mass spec analysis of the replica glues, and we found that the lime and alkaline environment that they were in really decomposed a lot of the material that was present. So because the historic glues also showed very little organic material, we think that there was probably a correlation to these cheese glues because there wasn't a whole lot left except for the mineral content. One exception was this particular vessel and this little punch bowl, and it's not a punch bowl like we would think of, it's a very small dish that would have been passed around full of glue. And full of glue. No, it would be full of punch, not full of glue. Sorry, bad mistake on my phone here. So what we did was we did a little bit of sample preparation, and we were able to find some resin acids and beeswax esters that were characteristic of a different recipe, and these appear to be resin glues. So we don't know for sure why Mary Ball Washington would have repaired her ceramics, but it seems more likely from the composition and sort of the way they look that it was probably more for display than it would have been for use. So they were probably just put up on a shelf to maintain their attractiveness on the shelf. So the ceramicist that brought us in on this project really loves this particular vessel because it's got this cherry decoration, and it's just so evocative of sort of the mythos of George Washington. And it turned out to be serendipitous that we're talking about this on President's Day weekend. So that's my story about DART mass spec. Thanks.
Hi, so I'm Christina Warner. I am an anthropologist and a biologist by training. Is my slides gonna, there we go, okay. And I work largely in the field of archaeological science, where I try to bring together as many different methods as I can to answer particular questions. And as an anthropologist, one of the things that interests me most are contradictions in human behavior. So I really like instances where people do things in really unexpected ways or do things that seem quite counterintuitive, because I think that's where you start to see some of the most interesting aspects of human culture at work in the past. And for me, one of the best examples of this is milk. So I think milk is absolutely fascinating. We produce a truly vast amount of milk today. So global production in this decade averages nearly 700 million tons of milk per year. And if you're like me, this is a really abstract number, and so I'm going to convert it into some more familiar units. That's the equivalent of over 275,000 Olympic-sized swimming pools of milk. If you're like me and have spent time in Massachusetts, that's 217 Walden ponds worth of milk. It's actually enough milk for every single person on Earth to drink a quarter liter of milk every single day. And yet, 2/3 of the world's population is lactose intolerant. So to me, this is a really interesting and puzzling contradiction. How did we take something that is a natural food for mammals and convert it into a food of really global distribution? How did we overcome the challenges of drinking this, and how did it become so socially widespread? Milk is actually consumed in lots and lots of different places in the world. So you're probably most familiar with it being consumed in places like Europe, but it's actually consumed traditionally throughout large parts of Africa, also South Asia, Central Asia, and even East Asia. And in each of these places, there have been numerous adaptations, both culturally and biologically, to utilize milk as a food system. And so one of the things, though, in my research, I really want to put this, push this back into the past and understand not just how it's distributed today, but how did this really arise? Where did it come from, and how did this technology spread? And so to do this is where mass spectrometry is extremely powerful. So what I do in my lab with my students is we work on recovering traces of proteins from the past, specifically from archaeological sites, from various different remains, such as dental calculus, which is calcified dental plaque or tartar, and also from different residues, to try to piece back together the diets of ancient peoples. So tomorrow I'm going to talk about some of our very recent work on looking at the earliest evidence of milk and dairying in East Asia.
So hello, I'm Elena Schrader, and I'm a paleontologist that actually looks at protein sequences preserved in fossils as an alternative way of getting at some of their biology and evolutionary relationships from morphology, which is how most paleontologists study ancient life, is through the shape of bones alone. One of the challenges with this, though, is that when you do protein extractions of modern bones, it's pretty simple. You do your extraction with a reagent, and you get proteins in solution. When you do this with some fossils, kind of this junk comes out that's basically the byproduct of decay called humic substances. And as you can see, the extracts that come out of those fossil bones can often be dark as coffee. And the color alone is not just a problem. Those humic substances in that extract can actually do things like clog your spray tips. It can break some of your spray tips, and it can suppress the ionization ability of your instrument, which is basically how the instrument is able to see protein sequences in the first place. So these substances, even if there's protein preserved in some of these fossils, the presence of these substances can interfere with the analysis of those proteins. So one of the things that I did, and it's not advancing, it's just this one, right? Can you press the next slide, please? So what I did is I developed a workflow that is basically geared toward removing large amounts of humic substances from some of the more problematic samples. Some fossils you get don't have a lot of humic substances, and some have a lot of humic substances. So I applied this workflow to a specimen of a moa, which is an extinct type of bird that's about 800 to 1,000 years old. And as you can see from the bone sample that I used, which is up in the corner, it is very, very, very dark chocolate in coloration, indicating that it's got a lot of humic substances in there. The workflow that I developed uses a couple of innovations that especially a lot of others had also contributed to extraction methods to take multiple aliquots of bone, take a lot of extract from a lot of bone, concentrate it into a small volume, and then remove those humic substances. So kind of what this looks like is you start out with kind of a dark extract that looks like sweet tea. You concentrate it down, which is all the way on the left there, and you can see it's this very, very dark, opaque, unusable extract once it's concentrated. Once it's filtered, you get a vast reduction in color. And then once you go through the stage tipping process, you get samples that are virtually indistinguishable from the blank controls. When I got there, I thought I'd actually lost all my peptides in the process. But it turns out, even though we had analyzed this specimen previously using a more traditional approach, and we'd only gotten 3 proteins altogether, this workflow enabled us to identify 18 proteins, which is 16 more than had been originally identified. Additionally, in the original analysis, we only identified collagen. In this analysis, we identified, I believe, 12 non-collagenous proteins that had not been described for this species. So basically, this workflow allows the analysis of really difficult samples that have a lot of humic substances, and it also allows the concentration of those extracts so you can get at some really low abundance proteins.
Thank you so much. So now we will open the floor to questions. Please raise your hand if you have a question. We'll bring a mic to you. Kindly state your name and affiliation before your question. So we have a mic coming to you in the 3rd row here. Just a moment.
Robin Williams, ABC in Australia. Christina, could you tell me, what did you discover with your analysing of milk that you didn't know before?
So the first step was just discovering that we could recover milk. It was a huge surprise to us initially when we were able to recover it from dental calculus, because as you imagine, milk is extremely difficult to track. It decomposes very readily. So before we applied proteomic techniques, the main method of detecting it was looking for dairy fats, which you infer from their isotopic ratios, and those are recovered from pots. One of the biggest problems we had is I've been interested for a long time in early dairying in East Asia. We tend not to think of dairying being present in East Asia, but for example, in Mongolia, dairy makes up the subsistence economy. It really is the foundation of the food system in Mongolia. And that makes a lot of sense because they're on the steppe. The steppe is mostly grassland, which is largely unusable, but ruminants can convert that into nutritious milk, which you can consume. But we have a huge problem in Mongolia, and that is because they're mobile pastoralists, we do not generally have sites, so we don't have the typical things that archaeologists look for or are able to find, like middens and trash heaps and households. We essentially only have mortuary archaeology. We have burial mounds. That's it. And they didn't bury anyone with pots. So one of the biggest problems is here we have one of the greatest dairy cultures in the world. In Mongolia today, they dairy more species of animals than any other country on Earth. 7 different species are milked to produce a wide variety of dairy products that have no analogs in a European system. And yet we had no ability to detect the prehistory of dairying there because we lacked all of the normal things we would look for. We lack the trash heaps to look for the animal remains. We lack the pottery to look for the lipids. And so we had to become really creative about going about trying to find it. And so this is how we developed using a proteomics-based technique, because what we have in Mongolia is an abundance of burials and an abundance of skeletons. By applying proteomics to dental calculus, to this tooth tartar, we were able to then turn this disadvantage into an advantage and actually detect at an individual level who in the community was consuming dairy products. And so we've been able to push this back to at least 1300 BC and are working on pushing it back even further in time.
Question in the back.
Hi, Dr. Warner, how precise can you distinguish the different types of milk, for like mare's milk versus cow milk, goat milk, et cetera? Can you actually do that?
Can you let us know who you are, too?
Oh, I'm sorry, Tom Garlandhouse, UC Santa Cruz, sorry.
Thank you.
That's a great question. Actually, this is where proteins are perfect because it's sequence-based. So when you're using a lipid-based approach, usually you can say dairy is present or not present. And then to some degree, you can distinguish equid milk from ruminant milk. But the real advantage of proteomics and using proteins is it's sequence-based. And so there are systematic sequence differences among these taxa. So we can distinguish cattle, sheep, goats, camels, horses. We can actually distinguish all of the major dairying animals. Yak and cattle are quite hard because they're very closely related, so there's only a few areas of certain proteins where we can distinguish them, but if we have good protein coverage, or peptide coverage of the proteins, we can distinguish them. So right now we can reliably distinguish cattle, sheep, and goat, and we've identified all 3 species archaeologically now. And we are now working on expanding that to the other dairy animals in Mongolia. So that's work in progress, but we will have exciting news soon.
Seems like you can learn a lot by looking at tartar. Teeth, what other questions about human behavior do you think you could undercover by looking at dental calculus?
So we found, a lot of people think I'm a little bit strange because I think the tartar in your teeth is one of the most interesting thing about a person. So please know when you go to the dental hygienist, you are just destroying the record for future archaeologists, and it's really tragic. No, there are so many things. I mean, calculus is amazing, and I'll show some images tomorrow. It grows throughout your lifetime. There's basically once you hit your 30s, you stop, from an anatomical, physiological perspective, you stop developing and you start degenerating. There's no part of you that continues to grow except your dental calculus, which will grow throughout your lifetime. And it grows incrementally and in layers, and it actually entraps aspects of your daily life. And so we find all sorts of interesting information. Most of the proteins that we find are actually not dietary related. Most of them are actually related to the bacteria that live on your teeth and your immune system response. So we can actually see kind of snapshots of your immune system at work preserved in the proteins of dental calculus. We also see lots of debris that comes in. So for example, those of you with seasonal allergies are very aware of the pollen that blows through. In the spring, we actually get little bits of tree pollen that becomes embedded in calculus of people, so little bits of that actually becomes embedded in your tartar. We also find evidence of craft activities, so we just recently published a study where we identified in the dental calculus of a woman buried in association with a medieval women's monastery lapis lazuli crystals. So this would have been used in association with a manuscript illumination. So you can actually reconstruct an enormous amount about a person from what gets preserved in sort of debris that passes through your mouth by eating and breathing and living. So it is very rich, and it contains a really high abundance of DNA, proteins, microfossils of plants, for example. We find starch grains, phytoliths, pollen, and then some of these more craft-related and kind of dusty craft remains.
Thanks. Makes me want to brush my teeth, but thanks.
Don't brush it. Don't brush it.
While we wait for the next question, the method that you developed, Elena, the new mass spec method, how long did that take you, and what was that process like?
So I actually tried a couple of other things and ran a couple of different, even of the workflow, I ran a couple of different versions of that workflow. One of the things that a lot of people use, especially in isotope analysis to try to get rid of humic substances, is sodium hydroxide. And one of the things I discovered is that you can only use that if you're using a certain type of procedure that demineralizes bone first. I also tried a lot of gel separation techniques. And serendipitously, I had kind of figured some stuff out about varying the pH, and then a week later, I thought I was crazy that I noticed that when I was putting formic acid in some of these samples to quench digestion, a lot of humics would fall out all at once, and I thought that I kept going back and being like, did I see that last time? And then 2 weeks later, a paper came out that was talking about this effect. And I was like, oh, so I'm not crazy. So it was kind of a long process of putting a couple of different things together and trying a lot of combinations of things to see what would work the best. And like I was saying earlier, when I got to that one part where the sample was very clear, I thought that I'd actually put it through such a hard process that I'd probably gotten rid of all my proteins. So to see 18, whereas I had only really expected collagen, because that's all that we got the last time, was kind of wild.
I think that's something that's actually really scary about our work, is that you're not really seeing it in action here, but the end stage of our work is an empty tube with invisible substances in it. And it really is kind of amazing. It's sort of a funny thing. You carry this sort of empty tube to go analyze it, but I really do think we're truly working in this sort of archaeology of the invisible and trying to recover all of these very small things that have been ignored and forgotten in the past, but can reveal so much information about everything from extinct animals all the way down to repair toolkits of the 18th century. So I think this is really exciting.
The kind of leap of faith that you take sometimes when you're like, all right, I've got this tube. It doesn't look like anything's in it. There can either be nothing or there can be everything. Like that moment standing in front of the mass spectrometer, crossing your fingers is something else, let me tell you.
That's one of the great things about DART is that we can see our samples. They're small, but at least we can see them. And to get data instantaneously is just so gratifying. Having done lots of LCMS, it takes a long time to get your data. And so when we can see something really quickly, even if it just gives us a snapshot of information, it's so great for my undergrad and grad students that they can just see their data pop up right away. It's really fun.
Hi, I'm Kathleen O'Neill, freelance. Ruth Ann, I was wondering about what this technique has allowed you to find that you wouldn't be able to find out otherwise, and what you had used in the past. Like, why does instantaneous results, why is that so important?
I think a lot of it is, can we answer questions quickly? So there are well-established techniques for studying things like dyes. I'm going to focus on the dyes because we've done so much of that. And most of these require taking a larger sample, extracting it into a solvent, sometimes removing that solvent and replacing it with another solvent again. So all of that has taken a lot of time. Then injecting it into the liquid chromatograph, separating it over the course of an hour or something like that, and then in the process of that coming through the chromatograph, actually identifying it by mass spectrometry. So that takes hours, days, depending on how you prepare your sample. We can literally take a little piece of fluff, stick it into the mass spectrometer, so small that you can't see it except under a microscope, stick it into the mass spectrometer and see the signal instantaneously. So this is something that we can sort dyes very quickly. And sometimes conservators have questions. What was the source of this red? Because if it's a plant red, we can't display this under certain lighting conditions. Whereas if it's an insect red, it's a little more robust, and so we can put it on display for longer with different lighting conditions. They're not asking me, what specific plant did this particular dye come from? They want to know very quickly, how can I display this? Can you tell me what this source is? So this is something that we can do without investing hours, weeks of time to do. Obviously, it doesn't give you the whole answer to the question, but it gives you an answer that might be useful depending on what your question is.
Question here.
Hi, John Carey, freelance. Dr. Schroeter, any new insights into moa biology from those extra 16 proteins?
Still working on it. One of the things that I'm very interested in doing is the original analysis of the MOA that we only obtained 3 proteins for. We'd use that data to actually look at a lot of post-translational modifications that had been preserved on these proteins. So proteins can be altered either biologically or diagenetically through basically geological processes while it's becoming a fossil. And the thing about how you search LC-MS data is that you need to search it against a database using software. And it does that by matching the masses in your spectra to the masses in the database. And therefore, for some programs, if you don't tell them what modifications to expect, it might not see it because you haven't told it what mass shifts to expect. So trying to get at what PTMs can be there, both biologic and diagenetic, is kind of important for kind of informing on how to search your data for a lot of other paleoproteomic studies. The original data that we did this on was the data that only had 3 proteins, so I'm kind of really interested to see if I can bulk that up even further with the new data.
There was recently a feature in the New York Times, and it was talking about ancient DNA, which is a newer process, people looking at ancient DNA from older specimens, and I think they were talking about maybe sometimes the tensions with archaeologists and understanding of different processes and events. Is ancient DNA information complementary to what you are doing? Do you work with ancient DNA geneticists on things?
So I do. So I run both a genetics lab and a proteomics lab, and I use them in complementary ways. So for example, in the study that I was mentioning, one of the things we were really interested in looking at was the introduction of dairying on the East Asian steppe, so in Mongolia, and whether or not that was associated with a migration event of people as well. Because we know during the Bronze Age, we get large-scale mobility and migration of herding populations, especially in the Western steppe. And when the dairy first shows up in Mongolia, it's associated with these Western animals. And so we wanted to know, was this process of introducing dairying one of a group migrating in and either intermarrying or taking over land, or was it more through a kind of cultural exchange? And so in this case, we use proteomics to identify the timing and the presence of dairying being performed and being consumed. And we use genetics to look at the population that was actually consuming the milk. And so what we found in that case, which was really interesting, was that this is essentially a local population that has adopted this Western pastoralist practice. There's a very small amount of admixture. We could tell that there had probably been some sort of intermixing between these two populations, but at a very, very low level. So the equivalent of, say, one great-great-grandmother or something like that in your background. But largely it was a local indigenous population that had wholesale adopted dairying. So I think there's so many ways in which you can put them together to tell a much richer story about what people were doing in the past and what sorts of interactions were like. So I think that they're complementary and should be used together.
So conversely, a lot of the specimens that I work with are beyond the range of DNA preservation. So DNA as a molecule actually has a lower preservation potential in terms of time than proteins. So one of the reasons that I use proteins is that it allows me to ask questions about species that have been extinct for longer than when we talk about things like human history. So one of the things I love about mass spectrometry in general, though, is that you'll notice a lot of we're using different methods and different approaches based on the kinds of questions we're asking about, and about the kinds of animals or artifacts that we're asking. So there are many different types of mass spectrometry approaches you can use that can be tailored toward the specific information that you're seeking.
I'd say there's two. It's interesting. They tell you kind of different things. So for example, a lot of the work I do is on food, and so if I'm just looking at food and I have DNA, it'll basically tell me the species. It doesn't really matter what tissue I'm measuring from that species. I will just get that species. This is where proteins are amazing because they're often highly tissue specific. That's how we can tell it's milk and not meat, for example. And so when you're looking at, there's certain questions for which proteins are much, much more suited. And the limits really right now on ancient DNA, just to put this a little bit in perspective, is there's this like 1 million year mark. It's been proposed that using the kinds of technologies we use today to analyze DNA, that there's a 1 million year limit, theoretical limit on how far back we could push DNA. But proteins extend much further back into the past.
Great. Question here?
John Kerry again. Any speculation on how the culture of dairying spread, if it didn't involve migration of people, and where it came from?
Yeah, so as far as we know, it originated first in kind of this really dynamic area around the Near East and Anatolia. And we're not totally sure if it first starts with cattle or sheep or goats, but it's one of those three animals, and dairying arises. What's interesting is that it actually starts being practiced at least 4,000 years before we have the first genetic evidence of lactase persistence. So this is really interesting. So most people here are probably familiar with the idea that there's certain genetic mutations that different populations around the world have that allow you to digest lactose as an adult. The actual ancestral state for humans is to lose this ability. It's the ancestral state for all mammals is to lose this ability as you age as part of the weaning process. But Europeans and some people from the Saudi Arabian Peninsula and three different groups in Africa have evolved mutations that enable lifelong lactase production and so are adapted to milk consumption. But what's so interesting is that we now know from ancient DNA that the earliest populations that were dairying actually lack these mutations, all these mutations. So we have at least 4,000 years of dairying taking place with no genetic adaptation to do so. It spreads really rapidly. It spreads rapidly both to Africa with cattle dairying and also into Europe, again with cattle dairying. And we're still trying to understand the timing of how it's going across through Asia, but it seems to be a Bronze Age phenomenon. And what's really interesting to me in Mongolia today, they've been dairying for at least 3,000 years, and today they do not have these lactase persistence alleles. So one thing that I find really interesting is that it seems to suggest that dairying itself does not require having these genetic adaptations, that there are other factors that are involved, whether that's cultural or perhaps involving the microbiome, that can also be adapted for milk digestion, and we are just starting to learn this now.
Question in the front.
For Ruth Ann, what are the handling considerations for dealing with a sample like the Washington China and putting it into your instrument?
So one of the disadvantages of using DART is that you ionize the molecules using a fast flow of gas. And we fortunately do this, we practice a lot with not precious materials before we get started. And I generally have my undergrads sort of try this and get a feeling for it. If you're not holding onto it tightly with a pair of tweezers or some forceps that you can lock, your sample can blow away. Just, that in general is a problem. I think the first time we tried running dyes, we watched our fiber be sucked into the mass spectrometer inlet as well. So there are a lot of things with just physically holding onto the sample. It can be challenging. With the Washington glues, we've also done some sample preparation. So if we run the samples just straight up, they tend to be mostly mineral content, which we can't see by DART. So by doing a little bit of sample preparation, so the advantage is you don't have to, but you can still do that and still get some different information. So introducing solutions is really trivial. Dip the closed end of a capillary tube into the solution that you've made, introduce it into the mass spectrometer source, and you instantly get signal. We don't see a lot of contamination from sample to sample. That's another nice thing, that we can do a lot of samples really quickly. I'm not sure if that actually answered your question, sorry.
Well, if there aren't any other questions, thank you so much to our speakers. That was tremendously interesting, and we appreciate your time. Thank you.